who's Napoca, is here. We have some support from US Air Force and Research Laboratory, and Dr. Bill Slicker, who is also one of my mentors, and he allowed us to work in US FDA for some time, and still that relation is going on. We have learned a lot. And of course, the Swedish Strategic Research Foundation that is dealing with military problems and also basic research. And obviously, I am coming from Uppsala University, that is established in 1477. So I will not have much time in detail to go through it, but I will try to show you the basic research that we are doing and fundamental principles involved. And since we are working with various US governments and Swedish government, I can tell that all the views that I am telling here is mine, and it has nothing to do with any organization mentioned above. What we believe in any kind of CNS injury, that blood-brain barrier is the basic of all kinds of diseases, because it is always normal if we consider we are healthy, and it is leaky in all kinds of neurological diseases. So we have to think that how stem cell can also acting to repair this blood-brain barrier, or it is passing through that, and what is its fate. And as you can see that when the capillary is leaky, it can influence neurons and also glial cells, affecting the whole system and our behavior. I give some of most credit to neuropathology is Professor Yngve Olsen. He was once president of International Society of Neuropathology. I started my work in 1988 in Uppsala University. And this is Dr. Sarvas Navarao. I'm not showing pictures to tell you something different, but I owe to this Professor Sarvas Navarao, who is given Bundestag in Germany by Helmut Kohl. At that time I was Humboldt Fellow there, and I learned a lot about electron microscopy of microvessel and many other things. At that time, in early time, when I was studying, then the former Prime Minister, the Honorable Rajiv Gandhi, was very much impressed with Sweden, because whatever the telecom revolution has come to India and all over the electronics and IT, I think he is the father of that in India. And he suggested that Sweden has a very high capacity of infrastructure, at least in medicine, so we should collaborate. Unfortunately, he was killed after that. Anyway, so we believe that this CNS insert opens the blood-brain barrier, and this is the basic region causing cell death. And I believe that the stem cells can do to alter many things, probably repair this, alter these pathways, and maybe helping. Regarding our involvement with nanoparticles, we came across when I was briefly in US FDA in Dr. Slicker's laboratory. Then with Syed Ali and my friend Sabar Hussain, they come up with the idea of nanoparticles and blood-brain barrier. So we did some experiments, and we published this volume in 2009. Then we also have a Journal of Nanoneuroscience by American scientific publishers, just to show that, and we have very high-profile editorial board. And we also have some support from Nobel laureate like Anders Shelley. The point is that we are doing something right towards that. And also we talk about the chemistry Nobel, telling our nanowiring things, and he believed that we are doing right. We met in Las Vegas recently. So we are trying to focus that nanodrug delivery can influence or can have better effect of the drug that is given normally. So the point is that we can use nanodrug delivery, and we are working together with Dr. Syed Ali and some other doctors. I will show you from the University of Fayetteville that we are making nanowiring of drugs and delivering this. So what is the role of nanoparticles in general in our lives? I mean, like many other countries, India is also one of them, where we have lots of air pollution around us. And if we have traumatic brain injury, or if we have a spinal cord injury in such an environment, probably the mechanisms of cell injury could not be similar. So this should be considered. And also it has implication in strategic point of view, that when the soldiers of different countries are placed at different places, they have different kinds of exposures. And naturally, when the injury occurs to them, the things are entirely different. This is just a review we have published about the role of not only nanoparticles but also disease processes. Because whatever we do in normal animals in laboratory, we have healthy animals. But together with Daphne Moresanu, we are working that what will happen if these animals were diabetic or hypertensive. So these are just to tell you, remind you that all these factors really influence drug therapy and its final outcome. So we have to think also in this way. So this is a long story, of course. I'm pretty sure I am encouraged by Dr. Slicker, who wanted to know more about toxicity of the nanomaterials. And I think they are doing a lot of work in US FDA. Then we are working, of course, with Chinese universities, and this is my friend Dr. Feng from Shijiazhuan. We have good cooperation there. And Dr. Shiqing Feng is here in Tianjin. We have very nice memory, and we are collaborating on many aspects of nano drug delivery in different aspects of also Chinese traditional medicine. This is the capital medical university, and these are all previously worked in Sweden. So we have a very long collaboration in different universities that is good for us. And recently, Dr. Shen is here. We went to Harbin, and we are establishing some good collaboration in neurology with that group. So making long story short, we are trying to test our hypothesis and trying to understand whether we are right or not. You know, United States has different government supports, and there is a National Innovation Summit occurring every year. So we tested our knowledge and submitted an innovation regarding cerebrolizing. Dr. Murasanu has introduced me in 2006. Because no single drug can be able to treat all kinds of things. And obviously a stem cell could be one of them. So we are using multiple neurotrophic factors, and it's a special drug called cerebrolizing. So we submitted some results to this National Foundation, National Innovation, and we are surprised that our work on neuropathic pain was given top 20% award. We are encouraged. So, and this was nano drug delivery of cerebrolizing. So we are very happy. I'm showing again some pictures to just show you some examples that how our soldiers are exposed to different environment. They are all breathing different kinds of nanoparticles. And this is a common situation in an area where there are so much violence. It could be street violence, it could be planned, or whatsoever it is. People are breathing different kinds of nanoparticles. So we must think at this time point of view that we should look neuroprotection in a very open and wide way. And that's why we are collaborating with Dr. Huang on stem cells. We are looking at whether nano delivery of stem cell could be even much more efficient, or some drugs like cerebrolizing can also affect the stem cell viability or maybe potentiating the effects. That I think Dr. Morasanu will tell you tomorrow. This is Dr. Anders Shelley, and we are working on some kind of small CRF factors in treating Alzheimer's disease, and also he has some interest in nano drug delivery of those things. He works in Miami. So we try to use titanium nanoparticles, because this is an example of carbon nanotubes where you can fill the drug and inject them. And now we are working on neurotoxicity of carbon nanoparticles. I will not disclose some of the results because this is subjected to national innovation for this year. And so this is Dr. Wang, and this is Ryan. They are the people working on nano materials, nano wiring drug for us. And as you can see that we have very close connection with Dr. Bill Victor. I am very grateful that he is having his maiden visit to India. And I am also very proud that I got a certificate of excellence from FDA, but this is not I am praising myself, I am telling that this shows that the works we are doing is good. Now coming to spinal cord injury, it is the very old problem and is still not solved yet, because we do not know that how a spinal cord injury can be solved, and I am showing you that even the 20th President of the United States could not be saved at that time because he has a spinal cord injury. And here, Dr. Sandberg is another key person, Dr. Huang and my other friends. They are working very deeply. We are very grateful to that. So what we look, neurosteratology, neuroprotection, neurodegeneration, probably they have many things in common. Coming from basic laboratory, I would like to study or focus my research on drugs, and as somebody here said that cells are also drugs. So briefly, I will tell you a few examples. What we believe, this is a very old philosophy, that whenever we treat patient, I think we should treat to the root cause, not the symptoms. And that is probably one of the main reasons that our many clinical trials fail. Dr. Wise Young is here, and I think he was actively associated in early 90s, naloxone trial of a spinal cord injury. Dr. Ferrin was there, and this drug is very good, very potent, but it failed in clinical trial. So these are the biggest problems. So I concentrated my research around the blood-brain barrier because we still consider that this is the gateway of neurological diseases. And Milton Brightman from Bethesda NIH first time showed that lanthanum when injected, it is stopped by tight junction. So this is one of the earliest pictures, the anatomical site of the blood-brain barrier, and it is open in all kinds of neurological diseases. This is the picture we replicated in our Swedish lab and showed that lanthanum is not leaking in rat spinal cord in healthy conditions. We still want to see what is happening when there is injury. Can many cells or drugs can pass from brain to the blood? Because this is also could be a brain-blood barrier is broken down. I do not have time to show some experiments. Then we are studying different angles of the spinal cord, and this is Dr. Eric Stahlberg. He has the credit to first single fiber EMG recording and assessing a spinal cord injury. I have the honor to work with him briefly. And we have a model of a spinal cord injury in which we are interested in gray matter lesion that is not involving white matter. And as we know that in clinical cases, we cannot find two similar types of cases. So that is one of the reasons that we are doing some kinds of mistakes or understanding the patient's conditions. And here it is just to show that one of the common causes of spinal cord injury is motor vehicle accident. In our animal model, we have applied different kinds of drugs, either topically because the concentration of the drug could be higher, because if we use some drugs it is diluted, or due to swelling or edema the drug is not reaching to the injured tissues. And here you can see that some of the drugs are doing excellent when applied topically. Pathologically, the picture is like this, and that can be reduced with drug treatment. My interest is that lanthanum is leaky, even the tight junctions are quite intact. This is an example in laboratory studies. And of course, there is a good relation with edema formation. Neural cells are also injured after a spinal cord injury, so as glial cells, massive upregulation of glial cells. So we have to not only see neuronal cells but also glial cells and axonal damage. This is an example of axonal damage. We are interested at electron microscopy, and here you can see that myelin vesiculation is principally responsible for most of the spinal cord injury cases, and there are also differences in different areas. I'm not going into details, but we can see that there are various factors are involved, neurotrophic factors and stem cells alike. When we have given a treatment that is BDNF and GDNF combined instead of BDNF, the cells look better, the spinal cord looks better. So this was our first interaction with combination therapy, and after that Dr. Murasan came and he said, well, you have done BDNF and GDNF, we have already a commercial material available that is called cerebrolizing, why not we should try that? And these are only telling that at a certain time point, if the combination is given, it is good, but not after. But we also observed that IGF-1 could be better than BDNF at the same time. And as you can see that there is a crosstalk between nitric oxide synthase when we have applied BDNF or IGF-1, the spinal cord nitric oxide synthase expression was down-regulated. So whenever we treat any drug or chemicals, it is working on various aspects because this is in vivo, it is not a cell culture studies. And we have also tested at electron microscopy that these drugs are able to reduce them. Okay. We can also see that when we apply the same kind of things, we can also downward regulate hemoxygenase. Then we are working in collaboration with Dr. Huang in Beijing and Sweden, and I will show you some of the examples very briefly. Here we have used different type of combination, and I can only tell you that the combination of different types of neurotrophic factors shows the best result after injury to the spinal cord. And these are the same thing. Water content is reduced. Blood flow is also increased by multiple combination. Neuronal damage is reduced by multiple combination. And this is just to show that hemooxygenase expression is also down. These are the pictures. And the multiple combination is reduced hemooxygenase expression after a spinal cord injury. So the point is that we have to address neuronal injury with multiple dimensions, with different types of treatment and combinations. So what will happen if we try stem cells also together with some of the drugs? And for example, showing this, the role of calcium nimodipine is the best, but we cannot use nimodipine because the clinical trial failed. It is showing so much high potential that it cannot be used. But since we are basic researcher, I can show you this picture that nimodipine has the best effect after a spinal cord injury. And they are also shown at the electron microscopy level. What is the relevance towards animal research to human? This was a very important question, and therefore we obtained some samples of human spinal cord injury from University of Perth, Australia, that is Byron Kakulis, who is a leading neuropathologist. And you can see that 6 to 8 hours after human cases of motor vehicle accident, there is a leakage of albumin. It means that blood spinal cord barrier is broken down in these cases, and that could be responsible for pathological changes. So again, we believe that blood-CNS barrier is very important. And I told you about cerebrolizin. It has different type of mechanism of action. I do not have time, so I will pass it quickly. And this is an ampoule, and we have some example of previous published paper. It is given intravenously. So what we did, that we also nanowired cerebrolizin and given in 2 different models of brain injuries. This is an open brain injury, not at all limited to the clinical, but this is concussive head injury, quite similar to human cases. And this is the result of open brain injury where we can have massive edema and leakage. If we give, for example, here nitric oxide synthase antibodies topically, we can see the picture is entirely different. So drugs are working in that way. But we have to look into different kinds of things and understand from our mistakes and laboratory investigation. So then we use the other part we have used is the animals were intoxicated with silica dust, because this is quite common for the military involved in desert. And as you can see, that silica exposure increases much more damage than the untreated one. This is also an example of tissue loss that is severely in silica treated groups. Then we have given nanowire drugs of different combinations, because we wanted to test whether nanowire drug or the drug itself is doing. And this is an example that we have given a compound that 5 minutes after and 1 hour after, it has reduced the changes in untreated group. So they have also, time factor is very important. And you can see that this is also same here. The point is that if we are using 3 or 4 different type of drugs and we are nanowiring in same way like stem cells or anything, can we have similar effects? The answer is no, because only good drugs can produce better result on motor scale, but some of the drugs, they are not working at all. So the drug is also important, not only the nanowiring technique. And this is an example here shown at different ways that the certain drugs are very good, they come in this area, and certain drugs could not do better, and some of the drugs are in between. So the drug selection is very important, not only nanowire technique. And this is an example here that how nanowire drug delivered can induce neuroprotection as compared to the untreated one. When cerebrolizin was delivered nanowire, you can see the myelin is the most better protected as compared to the other drugs given here like endomethacin as compared to the untreated one. And this has to do with heat shock protein, because the stress factor is very important. When we have given cerebrolizin, the stress reaction is much more less as compared to the untreated one. And so we are working all the time on edema and blood-brain barrier, and therefore we should have support from different kind of politicians, and I'm very happy that the chair of Rajiv Gandhi Foundation, Sonia Gandhi, is supporting our research from the very beginning. And also we have different supports. Our former Prime Minister from Sweden, Göran Persson, is very active in research and still involved. Our Swedish Education Minister is supporting our research. And not only that, our former President of India, Dr. Kalam, has great interest in nanoparticle research, and he wanted to make bridges with different countries. So these are some examples. Briefly, in 5 minutes I will conclude my talk, that nanowires can be, stem cell can be nanowired. That's one thing. And they can grow better if on different kind of nanowires. These are the examples that nanowired stem cells can survive a bit longer than non-nanowired, and you can see here their growth is quite good on nanowires. This picture you can see. But on the other hand, nanowires can also kill some cells at the cellular in cell culture. We are using this kind of nanowiring, titanium nanowire, and using different kind of drugs, and the material is quite stable. I will not go into detail in these things. It is the same thing that we have nanowired cerebrolizin against other methamphetamine-induced changes, so I will skip that and only show that if methamphetamine is given in diabetic groups, it produces much more damage than the methamphetamine alone. And when we have given nanowired BDNF 60 minutes after methamphetamine, results are better compared to the BDNF alone. And we are also working on hyperthermia, and here was YGN, a nice memory from Jordan. And here you can see when stem cell given alone and nanowired stem cell given, the pathology is much more clear than before. And these are the example: heat stress with diabetes, heat stress alone, and nanowired stem cells and stem cell alone can protect. The idea is nanowired stem cells have better effect than before. And these are some graphical example. Nanowired stem cell is better than normal stem cell in our model of neuronal damage. The same thing, they are reducing blood-brain barrier permeability much better than stem cell alone. And we can also see here at the electron microscopy, nanowired stem cell is preventing myelin damage much better than a stem cell alone as compared to the untreated group. So here I stop, and I am grateful to different kind of supports that we receive. And this is a huge collaboration. We have support from various organizations, my colleagues, and thank you very much for your kind attention.
Thank you very much. If there are questions, please.
Yeah, please. The mic, please. Yeah. Keep behind you.
Hi, thank you very much for your excellent lecture. I want to know that from your research, the combination of the neurotrophic factors maybe have good results. How to evaluate edema after treatment in spinal cord injury? Can you give us some suggestion in detail?
You mean you wanted to know how we have evaluated edema formation after a spinal cord injury?
Yeah. Combination the BDNF and the GNT.
Yeah. Actually, edema is accumulation of water inside the cells or inside the nervous system, extracellular or intracellular. And the simplest way to measure water content. So you take out the tissues of roughly similar size, sample size. Because if there are gray matters more, white matters more, it can vary the basic aspect of water content. Several cortex has one water content, different campus has different spinal cord as well. So you have to is roughly equal. Like 35 to 155 milligram. Then you do the same thing. Of course, when you have injury, the rate will increase, because of the water. And then you have the wet jet immediately taken after the experiment. After that, you put to take out the water, normally at 90 degrees Celsius in an oven for 72 hours. So you see that last 3 measurements of dry wet become constant. That means you have evaporated the water. And then simple formula, dry wet minus wet wet divided by 100, you can have the water content. This is one method. The other method that was very common is the gravity measurement. And in our laboratory with solar, together with Dr. Olson, a paper was published in Academic Pathologica in early 1990 that you can improve this gravity preparation. So you have a column and you drop the tissue, and naturally, according to the gravity, the tissue will floating at different levels. You can also calculate that. But after that, some people are telling that water content measure is still one of the finest measures to do edema. That's the biochemical estimation. The second thing that edema you can see is histopathologically, and there are many neuropathologists here that can identify edema. And if you go at electron microscopy, not only will you get extracellular edema, but also intracellular edema. But if you just want to measure edema, water content changes is very important. And then there is a formula of calculating volume swelling that is developed in early 1949 by Elliott and Jasper in American Journal of Physiology. So he has given a formula that is called percentage of swelling. Normally 1% increase in water can give you 4% volume swelling. So there are well-established methods in the literature, and I think you can follow for any kind of edema measurement in any tissue.
Do you use the MRI, for example, the functional MRI?
Unfortunately, our animal experiments and functional MRI are not allowed due to technical things. I do not have permission to do that.
Doctor, for multiple sclerosis patients, one doubt here. Hello?
Yeah, please.
For multiple sclerosis patients, the myelin sheet get destroyed.
Yes.
So for that also, since you are doing research in stem cells,
Yes.
so that therapy, is it really works for MS patients, multiple sclerosis patients, that titanium and all which you are talking about, can it be really useful for these patients?
Well, thank you for this question. I'm not working directly in this procedure, but I know very well in my early literature survey that in multiple sclerosis, blood-spinal cord barrier is broken down, and there are good number of papers published in early 75 and 80s in histopathological journal. So one of the idea is also in multiple sclerosis that you have to reduce demyelination, and any drug that can reduce demyelination, probably it will also help in multiple sclerosis. That is my point of view, but I'm not doing directly any kind of research in this area.
Okay, any more questions or it is time for lunch? Yes, one more question.
I have one very short question, probably just a conclusion of your presentation. Could you tell us very shortly what is the supposed mechanism of which is behind the superiority of this non-wiring growing stem cells comparing with normal stem cells? You showed that you grow these cells in this microenvironment, and there are some penetration of these nanoparticles into stem cells. So it is just a problem of easier penetration of the drug into stem cells, or there provoke some changes, plastic changes in the nature of stem cells growing in this environment.
Okay, I got it. So your question is that how and why nanowired cells or drugs can have better effect than without nanowiring?
How they affect the stem cells' nature, or at all, because there are some connections.
Okay. My short answer is that I do not know the mechanism that how it can change stem cells or drugs. But in general,